Okay, I have another question here. It says a cyclic reaction pathway requires that the starting material be regenerated and available at the end of each cycle. If compounds from the citric acid cycle are removed to be used as building blocks in a variety of metabolic reactions, why does the citric acid cycle not stop? Okay, I may not have talked about this, but it is true that intermediates from the citric acid cycle are drawn off. And they're drawn off quite often in quite a different variety of ways and quite a different variety of processes. And oftentimes it's to synthesize things like amino acids. Okay, so yes, things are being drawn off from the citric acid cycle. Intermediates are being lost. But the answer to this question is that yes, while things are being drawn off, the intermediates are constantly replenished as needed by reactions that lead towards the citric acid cycle. So what you're talking about is reactions that move away from or lead away from the citric acid cycle, but there are also reactions that lead towards the citric acid cycle. Okay, and those reactions can be quite useful in regenerating these intermediates. And one of these reactions is the conversion of pyruvate to oxaloacetate. And you might remember this, which is catalyzed by pyruvate carboxylase. You might remember this from my gluconeogenesis videos where I talked about this reaction. This was essentially the first reaction in the gluconeogenesis pathway. And anyway, it's quite useful in regenerating oxaloacetate. And as we know, oxaloacetate is the cyclic part. That's what makes the reaction cyclic. We regenerate oxaloacetate, the 4-carbon oxaloacetate molecule, at the end of the citric acid cycle. So this is essentially another way of making oxaloacetate, okay? So we have plenty of it available. And this is the actual equation, pyruvate plus carbon dioxide, ATP, and water makes oxaloacetate, ADP, and inorganic phosphate, and 2 hydrogen ions, or 2 hydrogens, or 2 protons, rather. And different metabolic pathways then work together to maintain the balance, to incorporate, to balance the appropriate concentrations of all metabolic intermediates required by the cell. So essentially what you have here is that if one set of intermediates is being drawn off, to be used in a biosynthetic pathway, then there's other pathways, okay, that are making up more of that particular intermediate, so that there's a constant supply, so that the concentrations are maintained, and that whatever the cell needs, the cell can have on a dime, essentially, immediately, is what I mean. So that's I thought this was another interesting question.